Hello everyone. Some people have asked, can we have a video introduction to the ellipsoid localization microscopy method for measuring bacteria spore coat structures? Yes, the answer is yes, you definitely can. Now, by way of introduction, you probably know that conventional optical imaging is diffraction limited, which means that if you have an object with a small size, like these bacteria spores, then the image that you get is blurred and diffraction limited, and features smaller than about 200 nanometers cannot be accurately distinguished in a conventional optical image. You possibly also know that various super resolution microscopy techniques such as ELM, or ellipsoid localization microscopy, do exist, which allow us to measure feature sizes much smaller than 200 nanometers, which is what we need to do if we want to look at the bacteria spore coat. So in this talk, I'll give an outline of the method, and you can read more details about this method in the biophysical journal paper. So the basic idea is that we have a bacteria spore, which is a fundamentally interesting structure. This is a bacteria spore, and if I put it into some boiling water, then it thinks it's having a nice warm bath, and when it comes out of the water and you put it on some food, it thinks great, and it begins to eat. So these things are very interesting just because they're very tough, and part of the reason they're tough is because they're protected by this thin grey armor coat, which has about 70 different proteins in multiple layers. And because the coat makes the spores tough, we'd like to know exactly how the coat works. But we can't do that using conventional optical imaging because the coat is only about 100 nanometers thick, or a bit less. So that means that we can't distinguish with conventional optics the order of different protein layers that make it up. But we might be able to do that using super resolution microscopy, and we have been able to do that using ELM. Now, what are we going to do? Well, we thought this structure, the protein layers might be 10 or 20 nanometers apart, so we might like to try single molecule localization microscopy, which should have the resolution to see these layers. Single molecule localization is very simple. The idea is that a fluorescent molecule is essentially a point source of light, because it's much smaller than the diffraction limit of optical resolution. That means that the image of a single molecule is a diffraction limited blur, and it's worse than that because the image is also pixelated by the camera, but that doesn't matter because if you know that the object you're looking at is a single molecule, then you can infer that its position corresponds to the center of the diffraction limited blur, and you can work out the center position of the blur very accurately depending on its signal to noise. And so if you want to know exactly where one molecule is on a glass cover slip, we can find that out very accurately. And you say, that's not very useful for looking at samples, because imagine I have a sample which looks like this. Imagine I have this important poster, but imagine it's only 2 micrometers wide. In that case, the optical image might be blurred and pixelated like that. So how could we use single molecule localization to read the important message? Well, the answer is if we have a photo-switching fluorescent molecule, we might be able to image a few molecules at a time and collect data like this. And by applying the principle of single molecule localization to each of these diffraction limited blurs, we can start to collect a list of all the molecule positions on the cover slip, and once we have an accurate list of all the molecule positions, then we can reconstruct a fluorescence density map, which is the super resolution image in localization microscopy. And it's much better than the diffraction limited image, and it would give us in spores the resolution that we would need to map out the coat layer structure. Problem is, that's quite hard. If we have a bacteria spore, we'd like to produce photo-switching fluorescence. But in fact, it's very hard to get sparse photo-switching fluorescence in a spore, I think for a few different reasons. One is that the spore is relatively big compared to some of the things which are looked at using localization microscopy, and there are so many potentially fluorescent molecules in a layer of its coat that it might be difficult to get sparse photo-switching fluorescence. Another reason is that the coat, which provides the spore with armor, is also fairly good at keeping out fluorescent stains, which you'd like to use to highlight a protein of interest within the coat. So I don't want to use single molecule localization this time, and I don't want to use this reconstruction technique, but I do want to use a different mathematical reconstruction technique to look at the come on, spore coat structure. And we want to use this ellipsoid localization microscopy technique. So the idea is very simple. Now our structure is, say, a spherical spore containing a fluorescent coat protein in this bright green layer. And we're not looking at photo-switching fluorescence, they're just all glowing or fluorescing normally on a microscope with some diffraction-limited point spread function. Because I'm saying the spore has a spherical geometry in this first case, I can work out an equation for the image of the spherical spore. And so the equation might say a particular spore of a particular size, radius A, with a particular microscope blur sigma, might look like, it might give you this image, diffraction limited. And that's a forward model. Given the structure, we get the image. And what we use in ellipsoid localization microscopy is once we have the equation, we do the inverse problem. We say we collect an image of a megatherium spore with a fluorescent coat layer, and it might look like that. And by finding the equation parameters which best fit the image data, we can infer very precisely the two parameters, the two main parameters of the equation. That's the radius of the coat layer that we're interested in, and we also pull out the blur radius of the microscope, which we might not need to know, but provides a check that we've worked out correct parameters. So that's the theory. Where do we get this equation from? So it comes simply from saying that a microscope is a linear shift invariant imaging system, and it's not as difficult as I've made it look. In a few lines of algebra, we get to an equation for what the image of a spherical shell should look like. And that's very nice. Is it right? So it is pretty good. So if we take that equation and we fit it to some actual spore images, we find that the pixel value or brightness of the measured images, shown with these blue lines, is very well fitted by the red line showing the equation fit. And the equation fit with only two parameters is so good that this suggests to us that we've got a model and some parameters which correctly describe the real structure. So with this, we can measure the exact radius, we think, of a single spore. Now, all we need to do then is scale up the process, and this is what our open source ELM software does. We say, given a fluorescence microscope picture showing a few hundred spores on a cover slip, we might ask the software to say, take this image, segment the image to approximately find each of the individual spores, and then for each candidate, fit the equation to the image data, and then tell me a list of the spore sizes. That's very useful. But we can do a bit more than that. We can also say, once we have this list of exact spore sizes, we can feed the structures back into the equation for the image data, and we can remove the effect of diffraction limitation, and we can make these reconstructions which show a visualization of what the structure would have looked like if it was imaged on a microscope with a better than diffraction limited resolution. That's very nice, but actually what's more useful is to, well, we could do that for an individual spore, so that we take the raw fluorescence data and we see the reconstruction is correctly aligned with it, but is better resolved. We can do that as well as for individual spores. We can do that for a few hundred spores with the same fluorescent coat protein, so we can work out the average layer of, say, the coat W protein and the average position of the coat E protein, and then we can produce the reconstructions with better than diffraction limited resolution, and we can distinguish the order which those proteins lie in within the coat. That can be shown nicely with this reconstruction. It's actually probably more useful to show it as a bar chart, where each bar shows the average position of the protein found in about 200 spores, and the confidence interval represents our uncertainty. The error bars represent our uncertainty in where the position of the protein is in the average spore. And if we do that, we found that our measurements were consistent with previous biophysical data with which some proteins were known to be in the inner coat and some proteins were known to be in the outer coat because they were more accessible for staining, for example. And our measurements were consistent with that, and we found that in the Megatherium spore, this coat E protein layer was found sort of as expected in between the inner coat and the outer coat, sort of expected because the coat E we thought might have a morphogenetic role, so a structural role connecting the inner coat with the outer coat. Good, so we can measure, we can map the structure of roughly spherical spores using this method, and I was very pleased with this. But we just needed to show what was the accuracy of this method. Well, we looked at a sample of, say, 200 spores on a cover slip, and we found some distribution of radii for a particular protein layer. This is for the SLAL protein layer with a GFP fusion onto it. And if we've repeated that for another batch of 200 spores, we found that the average protein layer was reproducible experimentally to better than 10 nanometers precision. That means that if we looked at two different proteins, which were separated in the case of coat G and SLAL by about 40 nanometers, the precision was good enough to say that they were definitely in a different place with the coat G further out. We could measure that experimentally, and we've also done some theoretical work to demonstrate the accuracy of the method. This plot shows that when we simulated using a realistic image simulation, the images of spores with an exactly known ground truth radius, then the dark blue line would show us the true radius which we'd like to recover, and these different markers show the radius which was recovered by different mathematical inference techniques. The bottom line is that using the best version of the ellipsoid localization microscopy, we recovered the green or light blue markers, which is to say we recovered the protein layer radius to within 15 nanometers without a calibration, or within 5 nanometers with a calibration. And the 5 nanometers left over is a random error, so really we can do better than about 10 nanometers in terms of locating the position of a single protein layer. That's if we have nice data, which means a nice bright image, no overlapping spores, and the real shape of the spore really is an exact sphere. So this is a theoretical study which shows we have about 10 nanometers precision, and that goes along with this experimental study where we could reproducibly locate a particular coat protein layer to within 10 nanometers of where we found it the last time. Okay, so we think the accuracy is very good, and I was happy with that. And then my colleague said, well, it's very nice, Eric, but you know that most spores are not spheres. They actually have an elongated, he said, almond shape. And he said, is that a problem? And I said, it's not a problem, it just makes things a little bit more difficult. We can do the same thing that I've talked about for spherical protein layers if we have elongated spores, provided we have a new model which describes the image of an elongated spore. And to get this model, we say, in mathematics, an elongated almond shape is roughly an ellipsoid, or precisely a prolate ellipsoid of revolution. And so the question is, can we get an equation for the image of that sort of fluorescent shell? And the answer is it's a bit harder, but yes, we can, given this structure where we have now not just a layer radius, but we have two axis lengths and an orientation. We can work out some sort of equation to represent the image of this object, and we can find that our equation for the image of an ellipsoidal shell can be a good fit to a measured image of an elongated spore like Satilis with a fluorescent coat protein. and our model is such a good fit that we think we can recover the axis lengths of the structure, and we can talk about that in terms of an equivalent radius. That's the radius of a sphere of the same volume as the ellipsoid which we've fitted to the image data. And so does this method work? So we can start off, how does this method work? We can say here's a fluorescence micrograph which we've measured on the left for a spore, and we can start off on the right with an approximate guess to what the parameters of the equation should be, and we can iteratively fit by gradient descent or random search, we can eventually find the equation parameters which best fit the image data. And once we've got those equation parameters, then we're back to the same place we were before with the spherical spore reconstructions. We can say, here's my shell, here are the parameters of my shell, and I can do this now for an individual spore, and I can see on the left that my blue pixel values in the measured image data are very well fitted by the red pixel values in the fitted model. And I can do this for lots of spores spread out on a cover slip, and then I can produce my reconstruction in which I very accurately measured the location of different protein layers in the ellipsoidal spores. And then I can plot a bar chart which tells me in Satellis, sure, we have SLA-L as an inner coat protein, we have coat G quite a bit further out, coat Z a bit further out still, and in my reconstruction I can map out where those proteins are. Okay. So that was the basic introduction to the principle of ELM, and you can get the software from our group GitHub site. So you go to www.github.com/quantitative imaging, and you can find the ELM software with its instructions for how to use it, and some illustrations of what the data analysis process should look like. There's some sample data for you to test this out on, and some instructions on how we think you can collect nice image data for putting into the ELM. Okay, so that's an introduction to the ELM principle as a inverse model. The software is open source, so if you try that, then hopefully you'll come back and I will put up another video showing some more advanced measurements that we've been able to make, such